Assalamu alaikum. So we were studying about the pancreas gland. Next topic in that line is the peripheral uptake of glucose. So we were studying about insulin in particular. As I've told you in the last lecture, pancreas has both endocrine as well as exocrine functions. And the endocrine glands, endocrine hormones released by pancreas include uh, insulin, somatostatin, and glucagon, out of which insulin is the hormone that is produced in the most amount. So let's talk about that. Insulin has basic effect on glucose uptake as well as lipolysis, proteins. So it basically increases the stores of energy in the body and it decreases the serum blood glucose level. Now there is a action of insulin, that is the peripheral uptake of glucose. It allows the tissues to take up glucose when at rest. Normally the muscles and other tissues that take up glucose when it is needed, when we are exercising or something like that. But at rest these tissues do not take up that much glucose. Now insulin, it compels them to take up and store glucose as well as fats. So glucose is taken up by peripheral tissues by facilitated transport. Passive transport not linked to sodium. Facilitated transport is whenever there is some energy being spent, some ATP being spent, or some factors that are being used for the transport of a specific compound inside the cells. Insulin facilitates this uptake in some tissues. Typically, the insulin receptor causes the insertion of glucose transporters in the membrane. So insulin receptor would insert glucose transporters in the membrane that would allow the passage of glucose from outside the cell towards the inside. Now, tissues that require insulin for effective uptake of glucose are, or the tissues, GLUT4 tissues which have this receptor are the adipose tissue, resting skeletal muscle, and cardiac muscle. So adipose tissue normally would not take up glucose, it would only uptake fat. Moreover, resting skeletal muscle would not take up this tissue. Just a second. Okay, so these tissues would not usually take up glucose normally. Cardiac muscles would not take up glucose without the presence of insulin or GLUT4 receptors. Resting skeletal muscle would not take up glucose. Although glucose can enter the working muscle without the aid of insulin. That is when we are exercising, walking, doing some activities, etc. The insulin is normally taken up by the skeletal muscles. They do not need insulin, but at resting phase only insulin can transport the glucose inside the cells. Moreover, adipose tissue. Okay, then there are some tissues in which glucose uptake is not affected by insulin. That is, they normally take up glucose, such as the nervous tissues, the neurons, etc., the kidneys, tubules, intestinal mucosa, and red blood cells. Insulin accelerates, but it is not required for the glucose uptake by liver. So nervous tissue, kidneys, tubules, intestinal mucosa, red blood cells would have no effect of insulin. They would keep on taking the glucose, uptaking the glucose normally in the presence or in the absence of insulin. Whereas in the liver, it normally takes up glucose and stores it in the form of glycogen. But in the presence of insulin, the glucose uptake is facilitated, it is accelerated at a faster pace. And insulin does this by enhancing glucose metabolism, not by inserting new transporters into the membrane. So glucose is metabolized quickly inside the liver. That's how the liver uptakes more glucose. It does not uh, uh, insert new transporter GLUT4 receptors into the membrane of the hepatocytes or liver cells. Now, what are GLUT4 transporters? There are 7 different glucose transporters called in order of discovery, GLUT1 to GLUT4. GLUT means glucose transporter. So there is GLUT1, 2, 3, 4, 5, 6 and 7. And GLUT4 is the transporter that is present in muscle and adipose tissues that is stimulated by the insulin. So a pool of GLUT4 molecules is maintained in vesicles in the cytoplasm of insulin sensitive cells, that is the muscles and adipose tissue. Let me explain this. So normally this GLUT4 is present in the cytoplasm of these cells. Now what happens is, whenever there is action of insulin, it increases the GLUT4 proteins at the cell membrane. So these proteins from the cytoplasm would move towards the cell membrane and embed themselves there. Now what would happen is, more glucose would be transported from outside the cell towards the inside, towards the cytoplasm, under the effect of these transporter proteins. So a pool of GLUT4 molecules is maintained in vesicles in the cytoplasm of insulin sensitive cells, and when the insulin receptors of these cells are activated, the vesicles would move rapidly to the cell membrane and fuse with the cell membrane. When insulin action ceases, the transporter containing patches of membrane are endocytosed, or again pulled back into the cytoplasm, and then they are covered in vesicle, and the vesicles are ready for the next exposure to insulin. So this is how all of this works. Normally these are present in the cytoplasm. When insulin comes, they move quickly towards the cell membrane and embed themselves there, transport the glucose, and when the insulin's action subsides, its quantity decreases, they would again be endocytosed or moved back towards the cytoplasm. This is how all of it works. Now in relation to potassium, insulin causes potassium to enter the cells, so it would cause hypokalemia with a resultant lowering of the extracellular potassium concentration. So there would be potassium moving from the blood or extracellular fluid towards inside the cell. So there would be lowered potassium in the blood, that is known as hypokalemia. Infusions of insulin and glucose significantly lower the plasma potassium level in normal individuals and are very effective for the temporary relief of hyperkalemia patients with renal failure. So normally what happens, if a patient of electrolyte imbalance comes to us in the emergency room or in the OPD, we conduct the tests and we find out that serum potassium level is increased exponentially. Normally the potassium should be between 3.5 to 5.5 milligram per deciliter or millimole per liter. If it is more than that, more than 5.5, 6.5 or 7, it is severe hyperkalemia. Now for the treatment of this, what do? We start giving the patient insulin and glucose. Now insulin would try to decrease the blood sugar level of that patient, but we are giving 5% dextrose or 10% dextrose solution along with the insulin, so to counter that insulin's effect. So glucose would be maintained at normal, but one more effect that would take place is the more potassium would be moved into the cells and the serum potassium level would start getting lowered towards the normal limit. So in hyperkalemia patients, an ideal treatment is giving insulin and glucose. The reason for the and hyperkalemia patient would often develop when patients with diabetic acidosis are treated with insulin. hypokalemia would develop if diabetic acidosis patients are treated with insulin. So what happens is, the reason for the intracellular migration of potassium is uncertain till now. If a patient has normal potassium level and we keep on giving large quantities of insulin to that patient, along with hypoglycemia, that patient would develop hypokalemia. So there is a condition which is known as diabetic ketoacidosis, which is a acute severe form of diabetes. That is a kind of an attack that takes place on the diabetic patient if insulin is not being given to the patient. In that particular disease, we start replacing the fluids of the patient, and moreover we give him or her insulin. But under the effect of this insulin, hypokalemia may develop. That's why along with insulin we keep on giving potassium to that patient. So why the potassium is moved inside the cell with insulin, we do not know this. It is uncertain, this mechanism. However, insulin increases the activity of sodium potassium ATPase channels in cell membrane so that more potassium is pumped into the cells. Now let's look at the mechanism of action of insulin by insulin receptors. So insulin receptors are found on many different cells in the body, including cells in which insulin does not increase glucose uptake. The insulin receptor is a tetramer made up of 2 alpha and 2 beta glycoprotein subunits, and this intracellular portions of the beta subunits have tyrosine kinase activity. So what happens is, these receptors, the insulin receptors, are widespread throughout the body. They are present in almost all of the organs, even those organs such as neurons which do not require insulin for uptake of glucose. They do have these receptors. They are comprised of 2 alpha chains and 2 beta chains. So these are 2 alpha chains and these are 2 beta chains, which are linked together by disulfide bonds, as we can see here, two sulfide disulfide bonds. Insulin attaches to the alpha receptors, act to the beta receptor, which have tyrosine kinase activity inside the cell membrane. This is the extracellular matrix, extracellular fluid, and this is the cytoplasm inside the cell. So inside the cell, tyrosine kinase would be generated, which would then have different effects in the cell. So this is how it works. Insulin comes and binds with the alpha receptors, which would then activate the beta ones, which would generate tyrosine kinase inside the cell. This would cause fat metabolism, glucose metabolism, growth and gene expression, protein metabolism, and glucose transport. When it acts on the glucose transport channels or GLUT4 receptors, more glucose would come inside the cell. So this is how all of this thing works. Now, how is the insulin secretion controlled? As I've told you many times over, all the mechanisms in our body are tightly regulated. When a hormone should be secreted or when it should be inhibited, it is tightly regulated by different processes of the body. So there are several factors which inhibit the insulin secretion, and then there are factors which stimulate the insulin secretion. So let's talk about the factors that would stimulate the insulin secretion. So what happens is, when there is increased blood glucose level, increased glucose in the blood, normally serum glucose level should be less than 126 milligram per deciliter, less than 126 at rest before meal. So fasting blood glucose should be less than 126. If it becomes 180 or 190 after meals or during fasting, mostly it increases up to 180 or more right after the meals. So what happens is more glucose would enter the beta cells of islet of Langerhans in the pancreas where insulin is being produced. So when there is increased intracellular metabolism of glucose, more glucose is present inside the cells. This increased glucose would be metabolized, broken down, and energy would be generated in the beta cells in the form of ATP. This ATP would go on to break down the proinsulin into insulin and C-peptide and release it in the bloodstream. Moreover, amino acids such as arginine also has the same effect. It stimulates the beta cells. Moreover, intestinal hormones such as gastric inhibitory peptide would also activate this beta cell, and glucagon's action would also activate this insulin secretion from the beta cell. On the other hand, when the glucose starts getting lowered, enzyme that is known as somatostatin is released from the pancreas that would stop the release of hormones from the beta cells. So somatostatin would stop glucagon as well as it would stop the release of insulin. Moreover, sympathetic innervation such as norepinephrine would stop the release of insulin from the beta cells, because during sympathetic activity we need more glucose to be available inside the blood so that we can produce more energy. So that's why insulin secretion would be inhibited by the norepinephrine. That is why norepinephrine, if released in huge quantities, would cause diabetes or hyperglycemia. So the most important control of insulin secretion is plasma glucose level. Above a threshold of 100 mg per deciliter, insulin secretion is directly proportional to the plasma glucose. So the higher the level of sugar glucose in the blood, the more the insulin would be released from the blood. For glucose to promote insulin secretion, it must not only enter the beta cells but should also be metabolized so as to increase the intracellular ATP concentration. So if insulin is to be secreted, the glucose would be taken up by the beta cells and glucose would be metabolized, broken down so that ATP can be generated, and increased ATP concentration would release the insulin. All the hormones or neurotransmitters named in the above attach to the membrane receptor of the beta cells or R receptors. In contrast, the metabolic substrates such as glucose and amino acids would directly enter the beta cell. So in the previous picture we can see that glucose and amino acids, arginine, is directly entering the cell membrane and then penetrating into the cytoplasm to release the insulin, whereas gastric inhibitory peptide, glucagon, these act via acting on receptors. Moreover, somatostatin and norepinephrine would also act on receptors. The next topic in line is glucagon. So glucagon is a hormone that is secreted by alpha cells of the islet of Langerhans. It is completely opposite to insulin. So all of the effects of insulin would be opposed by glucagon, and its effect would be complete opposite to the insulin effect. If you remember one hormone's effects, you can easily remember the others effects. So it is also a polypeptide hormone which contains 29 amino acids. Glucagon is also known as hyperglycemic factor because it increases the serum glucose level or blood glucose level. The primary target for glucagon action is the liver hepatocyte. It primarily acts on the liver. Skeletal muscle is not a target issue for the glucagon, in contrast to insulin, which primarily acts on the cardiac muscle, the skeletal muscle, etc. Glucagon mainly acts on the liver. It its main function is to increase blood glucose level. Uh, how is it able to perform that? Is via effect on different compound mechanisms, such as on the carbohydrate mechanism. It, by acting on liver, increases glycogenolysis, that is breaking down of glycogen, stored form of glucose in the liver. Moreover, it would stimulate gluconeogenesis, that is formation of glucose inside the liver. It would also have an effect on protein metabolism, that is, it would increase breaking down of proteins, proteolysis, and supplies amino acids to the liver for formation of new glucose or gluconeogenesis. This glucagon is also lipolytic and ketogenic. So it would break down lipids, fat, and it would generate 17 keto acids that would be then released in the urine. So why does it break down? Why it breaks down fat? To generate more energy so that more glucose can be produced. Other functions include positive inotropic effect on the heart, that is, the contractility of the heart be increased, and it would also stimulate the secretion of growth hormone, of insulin, and pancreatic somatostatin. So it would increase the release of all of these hormones: growth hormone, insulin, and somatostatin. Somatostatin and insulin would counter this glucagon, whereas growth hormone would be released so that the energy that is being produced by the glucagon, by making more glucose available, it could be used up for growth. It acts via GS receptor to activate adenylyl cyclase and increase intracellular cyclic AMP. Cyclic AMP would then activate protein kinase A, which would activate different enzymes of carbohydrate metabolism. Now, how do we control? So you need to just remember this: insulin activates tyrosine kinase, releases tyrosine kinase in the cell, whereas glucagon would release adenylyl cyclase, which would then release cyclic AMP. The release of glucagon, again the factors are completely opposite to that of insulin. Insulin was being released in hyperglycemia, it is released in hypoglycemia. So low glucose level in the blood is the most important physiologic promoter for the glucagon secretion. Elevated plasma glucose or hyperglycemia, when there is increased plasma glucose, would inhibit the release of glucagon. So decreased serum glucose level is its promoter and increased serum glucose level is its inhibitor. Amino acids, especially diabasic amino acids such as arginine and lysine, would also promote the secretion of glucagon. Thus, glucagon is secreted in response to ingestion of a meal rich in proteins. So whenever we take a proteinaceous diet, that is meat, etc., glucagon would be released in larger quantities. So it is stimulated, activated, the alpha cell of Langerhans, that produces glucagon, would be activated by a decreased serum glucose level, decreased blood glucose level, and amino acids such as arginine that would cause decreased intracellular metabolism of glucose and release of glucagon into the blood. Whereas increased blood glucose level would inhibit the release of glucagon. Insulin would act on its receptor on the cell membrane. Somatostatin would also act on the surface receptor on the cell membrane of the alpha cell and inhibit the release of glucagon. Now comes the last hormone of the endocrine pancreas, that is the somatostatin. So somatostatin is released by the delta cells of the islet of Langerhans. Alpha cells release glucagon, beta cells release insulin, delta cells release somatostatin. It is a polypeptide hormone that contains only 14 amino acids. On the contrary, glucagon had 29. Somatostatin is an inhibitory hormone that would stop the release of glucagon and insulin. So somatostatin acts locally within the islets of Langerhans themselves to depress the secretion of both insulin and glucagon. Somatostatin also decreases the motility of the stomach, duodenum and gallbladder. So the movement of food inside the stomach and the duodenum would be slowed down by a somatostatin. Moreover, inside the gallbladder, the movement of bile would also be slowed down by a somatostatin. Somatostatin decreases both secretion and absorption in the gastrointestinal tract. So it would just stop the gastrointestinal tract from doing its function. It would stop the glucagon and it would stop the insulin. It is basically the checkmate of chess. Okay, whenever there is this regulation of glucagon or insulin and the serum glucose level is going up and down, or this due to intestinal motility, insulin is being released and then stopped. This somatostatin is the main switch that would stop the working of all of these structures, so that all of the things can be then regulated and corrected. So regulation of blood glucose level is a bigger topic, and then comes the diabetes. We'll study it in the next lecture, inshallah. So I'll be sending this slide on WeChat, in the WeChat group. I've already sent the slide actually in the WeChat group, so you can download it from there. Okay, take care, Vachan. Allah Hafiz.